Good evening, everyone. So my next video, which I'm going to discuss with you, is on the topic types of enzyme inhibitors. And before I start my video, I want to request you, if you like this video and if it is helpful to you, kindly subscribe to my YouTube channel. That is Dr. Nitika Pharmacology Discussion. So now I'm going to start with types of enzyme inhibitors. First of all, I want to discuss whenever there is presence of enzyme in our body, the substance binds to it and this enzyme substrate complex is formed, and then the reaction occurs between enzyme and substrate, and it leads to formation of products. And the enzyme inhibitors, these are of three types, and these act on this enzyme substrate complex. They block this reaction from occurring. These can be the enzyme inhibitors can be of three types, that is competitive inhibitors, enzyme inhibitors, non-competitive enzyme inhibitors, and uncompetitive enzyme inhibitors. First of all, competitive enzyme inhibitors, they bind to the same site at which the substrate binds on the enzyme. Means there is competition between the substrate and the enzyme inhibitor uh, for the uh, attachment to the enzyme. So there is and this enzyme inhibition is surmountable. Means when we increase the levels of the substrate enzymes, the enzyme competitive enzyme inhibitor will decrease and substrate will increase, and that will lead to more reaction of the enzyme substrate reaction will take place and product will be formed. And this attaches only competitive enzyme inhibitor attaches only to the site at which the substrate binds. Means there is competition between substrate and enzyme inhibitor. Next is non-competitive inhibitor. Non-competitive inhibitor, it binds to the enzyme substrate complex also, as well as it also binds to the enzyme also. This non-competitive enzyme inhibitor, it binds to the allosteric site, that is at the site at which the enzyme does not bind to the substrate, but the substrate doesn't bind to this enzyme. At the different site, allosteric site, it binds to the enzyme, and it brings about the changes, uh, the changes in the structure of the enzyme substrate complex will be there, thus leading to inactivity of the enzyme substrate complex, leading to non-action of the enzyme substrate reaction. Means they bring about the change in the structure of this the enzyme. The non-competitive enzyme inhibitor binds to the enzyme substrate complex, and it produces the reaction in the enzyme substrate complex that produces the inactivity of this complex. Now this next is the uncompetitive inhibitor. It binds to the enzyme substrate complex. It has the affinity to bind to the enzyme substrate complex. It has high affinity to bind to the enzyme substrate complex, but it does not bind to the enzyme. It binds to the bound enzyme substrate complex, as the non-competitive inhibitor. It binds to the free enzyme also, as and also to the bound enzyme. This uncompetitive inhibitor binds only to the bound enzyme. And now I'm going to discuss with you Km and Vmax. Now, first of all, what is Km? Km is the substrate concentration in the body, in our body. It is the half of the substrate concentration. Sorry, the substrate concentration in our body, which produces half of the maximal velocity of the enzyme for the reaction to occur. Maximum velocity of the enzyme for the reaction, substrate concentration, it produces the half of the maximum velocity of the enzyme for the reaction to occur. And this is the Km. Vmax is the maximum enzyme velocity. Maximum is enzyme velocity for the reaction to occur. And what are the changes between Km and Vmax by these three types of enzyme inhibitors? First of all, the first I will discuss with the competitive enzyme inhibitor. It increases the Km and it has no effect on the Vmax. Why? Because it reduces the affinity of the enzyme, affinity of the substrate to bind to the enzyme. Like if there is in our bodies, suppose if there is five enzymes and sorry enzyme is there, and the five enzymes are there and five substrates are there, they will produce the reaction and the reaction will be produced, and that is 5 km, and this the km is 5. Now if we add the make the enzymes 10, and we add 5 substrate and 5 enzyme inhibitors, that is competitive enzyme inhibitors, that reaction will again be 5, not 10. The km will be again 5 because substrate enzyme will be 5 5, they will produce the action, and the 10 out of the 10 enzymes, 5 enzymes will be non-functional. So when we increase the substrate, like if we increase the substrate to 10, and enzyme inhibitors are 5 only, and enzymes are 20. So that will lead to increase concentration of the substrates in the body. So the affinity for the substrate, it reduces in case of the competitive enzyme inhibitors, but the substrates will be more in our body, they will be 10, so that will produce more enzyme substrate complex reaction. So the affinity in the body reduces, which leads to increase in Km. And the Bmax, it has no effect because it is surmountable competitive enzyme inhibition. So we can increase the levels of substrate. So maximum reaction velocity does not change. Now, secondly, now non-competitive inhibitors, they have no effect on the Km, but they reduce the Vmax because the action is in the enzyme substrate complex, and the enzyme, they will produce the physiological, the structural changes in the enzyme substrate complex, making the enzyme substrate complex non-functionable. The changes will be produced. So the Vmax will be reduced, but it has no action on the Kmax. Sorry, Km. The affinity is not affected. And non-competitive inhibitors I have discussed, and uncompetitive inhibitors, these are the enzyme inhibitors which bind to the enzyme substrate complex, and they reduce the Vmax also, and they reduce the Km also. They reduce the increase the affinity of the substrate to bind to the enzyme substrate complex, and they decrease the Vmax also, and they decrease the Km also. And the difference between the competitive and uh, non-competitive enzyme inhibition is that competitive is the surmountable enzyme inhibition and non-competitive is the unsurmountable enzyme inhibition. The examples are like an antagonism is the example is morphine and naloxone, and in the competitive enzyme inhibition, and in the non-competitive enzyme inhibition, the example is diazepam and bucoculin, which are used in the sedative as the sedative hypnotic drug, and morphine is the opioid analgesic drug. So they the bucoculin binds to the another site on the enzyme and produces the non-competitive enzyme inhibition. Thank you. That's all with the type of enzyme inhibitors. I hope you understood my lecture. And if you understood my lecture, kindly subscribe to my YouTube channel, that is Dr. Nitika Pharmacology Discussion. Happy learning. Be happy, be healthy, be blessed. Thank you. Stay safe. Happy learning.